Hello, my name is Priscilla Sano, and today my presentation will be based on intracellular compartments and protein transport, and my presentation will also be geared towards an undergraduate level. We have a few objectives that we will go over. Number one being the compartment importance, cell strategies for isolating and organizing reactions. We will have an overview of membrane-bound organelles and their functions. Then we will shift our gear into proteins and why transport is actually needed. 3 mechanisms of protein transport, how they know their destination, vesicular transport and docking, 2 secretory pathways, which are exocytosis and endocytosis. And within endocytosis, we will focus on phagocytosis and pinocytosis, and some research that has currently been done on compartmentalization and protein transport. The importance of compartments. So cells carry out numerous reactions at the same exact time, and that is because eukaryotes do have many different organelles that are found within them. All of these organelles have different functions, different tasks that they're performing pretty much at the same exact time. And thank God that these organelles have membranes that do surround them so that whatever reaction is being run is held intact within that organelle. If not, all of these reactions would be taking place in one central location, such as the cytosol, and that would cause chaos, and eventually cell death could actually occur. So intracellular processes occurring at the same time have to be separated. Cell strategies for isolating and organizing reactions. So number one, they aggregate the different enzymes needed to catalyze reactions into large complexes. And these complexes form large biochemical subcompartments with specific functions. Number two, they confine different metabolic processes and the proteins needed to perform them within different membrane-bound compartments. And this leads us to the membrane-bound organelles that we now know are found within eukaryotic cells. An overview of membrane-bound organelles. Prokaryotes are composed of one compartment and they are surrounded by a cellular membrane, whereas eukaryotes are quite different because they're very complex and they're subdivided by internal membranes with very different organelles. So each organelle has a specific function for the overall benefit of the organism. The first organelle that we will be looking into will be the cytosol, and the cytosol is the fluid mosaic that's found within the cell, and it is surrounded by a cell membrane. It is the site where metabolic processes take place. Organic and inorganic substances are found along with spare nutrients. The next one is the nucleus. It's the cell's organizer, somewhat like the boss that dictates everything that happens within the cell and within the organelles. This includes the arrangement of chromosomal DNA, how the synthesis, processing, assembly, and transport of macromolecules are coordinated and regulated. Now, the nucleus does communicate with the cytosol through the nuclear pores, and it is a continuous membrane with that of the endoplasmic reticulum, which we'll be looking at next. The ER is a major site of protein synthesis and transport. Protein folding does occur, lipid and steroid synthesis, carb metabolism, and calcium storage. Now, there are 2 types of ER. One is the rough, and the other is a smooth ER. The rough ER is termed that because it has ribosomes that are embedded within the entire endoplasmic reticulum of the rough ER. Now, the whole function of the ribosome is protein synthesis. So as it makes proteins, they're going to be delivered inside of the endoplasmic reticulum's lumen. Smooth ER is somewhat found in specialized cells. So here are a few examples of what smooth ER can do in these types of cells. It can produce steroid hormones in adrenal glands, endocrine cells. It detoxifies organic molecules in liver cells, and it can release and uptake calcium ions from the cytosol for muscular contractions. The Golgi apparatus. It serves as a role in intracellular membrane traffic. So it receives all the secretory material and membranes synthesized by the ER, and it'll modify them by glycosylation. Then it'll package and send them in vesicles to their correct destination. It's also capable of complex polysaccharide synthesis used for building cell walls in plant cells. So here we have a visual of a Golgi apparatus and a vesicle that is used for transport of proteins and other substances. Now, later on, we're going to talk about a process called endocytosis, but in endocytosis, certain things are going to be needed by the cell, and they're brought in through the cell membrane. The cell membrane will actually form somewhat of vesicle, and it'll pinch off inside of the cell. Then it'll be transported into an endosome. So the endosome will sort contents before they get to the lysosome, and it'll recycle some of those components back to the plasma membrane. Then it'll travel to the lysosome where it degrades and recycles cellular waste. Now, recent studies have revealed a lysosome to nucleus signaling pathway and a lysosomal gene network that regulates cellular clearance and energy metabolism. So we can't just look at the lysosome as kind of like a dead-end organelle, but it can now be viewed as a control center of cellular metabolism. Then we have the peroxisome. It enzymatically breaks down lipids and is involved in hydrogen peroxide detoxification. Now, evidence does support the view that the peroxisome can also function as an intracellular signaling compartment and as an organizing platform that makes certain developmental decisions from inside the cell. The mitochondria. The mitochondria is known as the powerhouse of the cell, so it's responsible for energy production in eukaryotes. Mitochondria can actually be found within plant and animal cells. Now, this whole process of energy production is run through cellular respiration. It can also synthesize phospholipids and heme, maintain calcium homeostasis, apoptosis activation, and cell death. I found this quite interesting because I didn't know that the mitochondria was involved in cell death, so I did a little bit of research behind that. So the mitochondria has pro-death factors that are found in the mitochondrial intermembrane space. So pores are formed in the outer mitochondrial membrane. This releases intermembrane space proteins and promotes capsase activation and cleavage of structural and regulatory proteins in the cytoplasm and within the nucleus itself. So this all eventually will lead to cell death. The chloroplast is the major conversion site of the sun's radiation energy to chemical energy that's usable by organisms, and this whole process is termed photosynthesis. They are also the place of synthesis of fatty acids in plants and biosynthesis of amino acids. Now, the mitochondria and chloroplast, although they're very different structures because they run two completely separate processes, they are somewhat similar in the sense that they do possess their own genome. So this suggests that they evolved from bacteria and they were later engulfed by eukaryotes, and eventually they came to a point where they lived in symbiosis, and this is the whole basis behind the endosymbiotic theory. Many organelles are held in their spots in the cell by microtubules of the cytoskeleton. The cytoskeleton has filaments that provide tracks for organelle movement and directing vesicular traffic between organelles. And it's involved in lots of different other things as well. So a couple of examples, it is involved in endocytosis. It is involved in cell division, where it helps to pull apart the sister chromatids to ensure that cells will have the same exact type of DNA or the number of chromosomes specifically. And it helps to adapt a cell with cell shape. So the cytoskeleton is a very versatile structure. Now shifting our gear into proteins, why is protein transport needed? So proteins are continuously going to be made by ribosomes in the cytosol, and they need to reach an actual location. So their destination depends on an amino acid sequence or a sorting signal. Some proteins will be excreted from the cell, and then others will replace degraded organelle proteins, and this is needed for cellular growth, division, and normal cell functioning. There are three mechanisms of protein transport, so organelles need to be able to transport water-soluble proteins across a hydrophilic membrane. Proteins from the cytosol are going to move to the nucleus through nuclear pores. So these nuclear pores do serve as gatekeepers, allowing specific macromolecules to come in. Proteins from the cytosol to the ER, mitochondria, and chloroplasts are going to pass through protein translocators. So protein will end up unfolding so that a translocator can guide it inside of the membrane. From the ER and from one compartment of the endomembrane system to another, they're carried by what's called a transport vesicle. So vesicles will pinch off the membrane of one compartment and merge with another. In this visual, it shows somewhat that. So we do have a cell membrane, we do have a vesicle that's carrying something foreign that was found inside of the cell. Okay, so what's going to happen is this vesicle will merge with the cell membrane, and then eventually the cell membrane will open up to expel the contents to the outside. How do proteins know their destination? So an amino acid sequence does provide that signal for the protein destination, and we just learned that was called a sorting signal. So the table shows two examples of signal sequences used to specify different destinations inside of the cell. Vesicular transport. Once a protein travels to the ER, it needs to get elsewhere, and the next destination is going to be the Golgi. So from the ER to the Golgi and Golgi to the endomembrane system, vesicles will be extremely important. Once a vesicle has been shed from the membrane it came from, it will be coated. Eventually, it will lose that coat so that the membrane can interact with the destination's membrane. Vesicular docking. So these are the steps that the vesicle will take in order to be able to merge with a receiver. So the vesicle is going to frequently move by motor proteins that move along the cytoskeleton. Once it gets to its target location, it's going to dock. Transport vesicles display molecular markers called Rab proteins on their membrane, and these Rab proteins are going to bind with tethering proteins on the target membrane. Matching the correct Rab and tethering proteins ensures that the vesicles reach a correct destination and there are no mistakes being made. And then snares on both the vesicle and the target membrane will also bind to ensure that there's a firm hold, a firm grip on that docking, and thus delivery of the contents will be the ultimate goal. Here we see a visual. Okay, so we have a vesicle that's being shed. Okay, the vesicle does have contents inside that need to be delivered to a target. So we do notice that it does have a protein coat. Eventually, once it does come apart from the membrane, okay, the vesicle will end up losing that protein coat. The vesicle will travel along a cytoskeletal tract by movement of motor proteins. Okay, now this vesicle does have a Rab protein and it does have a snare. So eventually, once it gets to its target, that Rab protein will need to bind to a tethering protein. And do notice that the vesicle and the receiver do both have snares. So eventually those snares will come together to form that tight grip, and it will end up merging with the membrane of the target to be able to deliver its contents inside of that target cell or target organelle. Secretory pathways, we have exocytosis. So vesicular transport is not confined within the cell, but it can merge with the plasma membrane to exude contents that are needed outside of the cell, and that's called secretion. Exocytosis contents can include proteins, lipids, carbohydrates, hormones, mucus, waste, and enzymes to be released outside of the cell and delivered to other places within the body. Proteins can be modified in the ER, further modified and sorted in the Golgi, and then a vesicle will take these contents to the plasma membrane. Here we see a visual where we have a vesicle that needs to transport a certain substance to get outside of the cell. Eventually, it'll merge with the cell membrane, and it'll open up to be able to expel those contents. The vesicle will merge with the membrane, causing it to increase in size for a split second, but eventually it will decrease in size as well because endocytotic pathways will return the contents inside of the cell. Now, exocytosis can also help to supply the plasma membrane with new lipids and proteins so it can make more of itself before a cell division, or to replace old proteins and lipids in cells that do not divide or will not divide. The next pathway is called endocytosis. So cells take up fluid, molecules, large particles, and other cells by this process. The plasma membrane will enclose the item, and then it will pinch off inside, forming a vesicle. It will travel to an endosome so that contents may be recycled, or to the lysosome where contents will be digested. Digested food items will be released into the cytosol for organelles to be able to utilize that energy. Within endocytosis, we have a process called phagocytosis. So the process involves cellular eating or ingesting large particles, but it is important for things other than nutrition. Phagocytic cells do ingest microorganisms. So what's going to happen is we have an alien invader, and then the phagocyte will form what's called pseudopods to be able to surround and somewhat encapsulate that alien invader. Now, once the microorganism is entrapped, then the phagocyte will actually bind with the lysosome for destruction. Now, these cells also serve to uptake dead and damaged cells. One cell that's very commonly broken down within our bodies is the red blood cell or the erythrocyte. We do know that the lifespan is typically about 180 days. So when those cells die, these phagocytes are very important to be able to pick up that cellular debris, recycle contents that can be used again, and get rid of other substances that cannot by lysosomal action. Now, these phagocytic cells are called macrophages, and another type of cell is a neutrophil. They're both different types of white blood cells that do help with protecting the body against alien invaders and foreign substances. The other process that's a part of endocytosis is called pinocytosis, and it does involve cellular drinking. So what's going to happen is basically the cell membrane is somewhat going to invaginate and entrap water molecules. Okay, eventually it'll pinch off and form a vesicle on the inside in order to be able to bring in that water content that the cell does need. So there is some current research that's being done. This one by Beals and Associates. There was an article written that's called The Artificial Cell, Biology-Inspired Compartmentalization of Chemical Function. So in principle, an artificial cell could act as a chassis for the integration and coordination of biological and biomimetic functionalities to achieve desired and useful properties. Also, understanding how to construct an artificial cell will lead to a deeper understanding of cell biology and living systems, and it helps to provide deep insights into the properties necessary for the origins of life. Some other research, this is on protein transport. It's by Mironov and Associates. They wrote an article called Models of Intracellular Transport, Pros and Cons. So this review describes the current understanding of Golgi function while highlighting the recent progress that has been made. It continues to discuss the questions and avenues for future research with regard to the models of the following intracellular transport steps. It does look at the vesicular and diffusion models, the compartment maturation progression model, and the kiss and run model. So in review, we did talk about the importance of compartmentalization, cell strategies for isolating and organizing reactions, membrane-bound organelles and their functions, why protein transport is needed, 3 mechanisms of transport, how proteins know their final destination, vesicular transport and docking, secretory pathways of exocytosis and endocytosis, and looking a little bit closer into endocytosis is phagocytosis and pinocytosis, and some research that has been done on compartmentalization of protein transport. I would appreciate any questions that you have based on my presentation. And then here we do have references and other resources that can be used in order to find some more information about compartmentalization and also with protein transport. Thank you very much for your time, and I hope that this presentation clarified anything that might not have been so clear previous to this. Have a great day.